to the last of the 2020 CIBM Breakfast in Science seminar series. It is my great pleasure to introduce you Lijing Xin, who is the research staff scientist at the CIBM MRI EPFL AIT section. Her talk will be on probing brain metabolism in vivo, insights from multinuclear magnetic resonance spectroscopy. Thank you, Lijing. The floor is yours.
Good morning, everyone. Thank you, Pina, for the invitation, also the introduction. So today I will first give a brief introduction about multinuclear MR spectroscopy, and then I will talk about two collaborative applications that we did use phosphorus and C30 MRs to address specific research topics. So these are different MR images from for assessing the brain structure and function. Despite the diversity MR imaging, the signal we observe originates from water. In fact, under the intensive signal of water, if we zoom in for 1,000 times, there are large amount of molecule information that we can measure with MRS. For example, here is the proton MRS. We can measure metabolites involved in neurotransmission, like glutamate, GABA. There are also other metabolites involved in brain energy metabolism, membrane metabolism, osmolytes, and antioxidants. And actually, there are many MR visible nuclei besides proton, like deuterium, carbon-13, nitrogen, oxygen-17, phosphorus. The fact that they are not widely used is due to their low sensitivity, leading in technical challenges. This low sensitivity originates from the three factors. The one which I just mentioned is the low concentration, which is 1,000 times lower compared to imaging signal. In addition, they have a low geomagnetic ratio and the natural abundance, which worsen the sensitivity. However, through X-nuclear MRIs, we can actually probe unique insights that cannot be achieved by MRI. Using different nucleus, we can probe different metabolic pathway in vivo and achieve both static and dynamic informations. This is one example showing here the energy metabolic pathway from glycolysis, TCA cycle, all the way to the electron transport chain, where C13, phosphorus, deuterium, and proton, also the oxygen-17 could offer the measurement of ATP level, metabolic rate of a TCA cycle, glutamate-glutamine cycle, oxygen consumption rate, chemical reaction rates for ATP production and utilization. So I will briefly talk about two commonly used nuclei. Basically, it's the phosphorus and the carbon-13. Using phosphorus MRs, we can measure high energy phosphorus compounds like ATP, phosphocreatine, and also the inorganic phosphate. Together, we can also measure the glycylphosphorethanolamine, glycylphosphorocholine, phosphoethanolamine, which are essential membrane phospholipid turnover metabolites. Phosphor MRS also offers the measurement of some physiological parameters, such as intracellular, extracellular pH, and the free magnesium concentration derived from chemical shift difference between resonances. Recently, the measurement of oxidized and reduced form of nicotine amide adenine nucleotide, the NAD plus and NADH, has been reported in cat and human brains. And this brings a lot of attention actually in the field recently, in the recent years, as NAD is a very important coenzyme involved in energy metabolism, all the way from the beginning of the glycolysis TCA cycle to the electron transport chain. The intracellular NAD redox states the NAD plus over NADH ratio regulates the energy metabolism. In addition, the NAD plus is also an important co-substrate. for NAD plus consuming enzymes that are important for many cellular signaling processes. And the change in NAD plus and NADH could implicate in mitochondrial dysfunction and neurodegeneration and also aging. So lastly, using endogenous tracer phospho-MRS can also measure dynamic information, including chemical reaction rates of creatine kinase and ATP synthase, which are the two key enzymes in neuroenergetics for supporting normal brain function. To measure the chemical reaction rates, a magnetization transfer experiment is usually used. The main idea is to perturbate equilibrium magnetization of one molecule nuclear. Since the perturbated molecules convert to other molecules due to chemical reaction, we can observe the transfer of these perturbation to the same nuclear but in their chemical products. In this case, the perturbation can achieved by either spin saturation or inversion. Here, I will do one demonstration for the saturation transfer experiment. So before saturation, we can see, although there's a chemical exchange happens, but we could not see the change in MR signals as the total amount of MR signal in each pool did not change. And now we add additional saturation pulse on ATP pool, for example. And this tends to actually equalize the probability of a spin energy states, and we start to lose the MR magnetization, in this case for ATP molecule. So through the chemical reaction, this saturation starts transfer to inorganic phosphate and the phosphocreatine pool and lead to the reduction of their MR signals. So how fast the signal decay reflecting the chemical reaction rates of creatine kinase and ATP synthase activity, the two activity here, then we can use a modeling by block equation to derive the relative reaction rates. So comparative phosphorus, C13 has only 1.1% natural boundness. This actually makes it worsen the sensitivity, but it is also not a bad thing because we can actually profit this property. By introducing exogenous C13 labeled substrate, we could easily follow the fate of the substrate to measure the metabolic activity all the way along the pathway. So C13 labeled glucose acetate lactate are commonly used substrate for C13 MRS experiment. Downstream, the label will arrive glutamate, glutamine, which are sufficiently high concentration that we can monitor by MRS. So recording the dynamic C13 labeling signal in glutamate and glutamine by C13 MRS, then further use mathematical modeling, we can actually quantitatively determine metabolic rates of TC cycle, glutamate, glutamine cycle for the in vivo system. Of course, this kind of approach could also be extended to other substrates and address their relevant metabolic pathway. So first, I will present a collaborative work with Nestle that we investigate brain energy metabolism and aging using phosphorus MRIs. Sorry? Using phosphor MRIs. So this work was recently just accepted in Frontiers in Neuroscience. So aging is associated with a declined mitochondrial function and a decreased cerebral energy metabolism, which are major risk factors in developing neurodegenerative diseases. So as we know, NAD plays an important role in energy metabolism and does reflect mitochondrial functions. So we use phosphoramers as a method of choice to investigate the impact of age on NAD redox status, brain energetics, and membrane turnover. Then we further explore the association between these components. So this study were conducted on 50 participants across a large span of age from 19 to 70 years old, with two main age group, a younger age group and an older age group. So we performed phosphorus MRS measurement at 70 with a surface coil dedicated for human occipital lobe. And then the major phosphorus metabolites, including energy metabolites, NAD content, and membrane turnover metabolite, were measured by pulsar-quiet sequence. And the chemical reaction rates for ATP production via the creatine kinase and the ATP synthase were measured by steady-state saturation transfer experiment with a scale. Reduction in ATP and also reduction in phosphoethanol. The reduction of ATP with age suggests a brain energy hypometabolism during brain aging. We also observe in the older group increase in phosphocreatine, ATP production by ATP synthase, and membrane degradation reflected by phosphodiester. So phosphocreatine is the energy buffer to maintain ATP level. So this increase in phosphocreatine suggests that the age the brain could not actually sufficiently maintain ATP level through this pathway. So the increase in membrane phospholipid breakdown and the reduction in membrane synthesis in older age is also consistent with previous results that suggesting it may be the consequence of oxidative stress that lead to NAD damage and cell death. So based on correlation results, we were able to constitute a phosphometabolic network that's linking age, NAD levels, redox states, energy productions, and also the membrane phosphometabolism. So here the negative correlation showing in blue and positive correlation showing in red, and the thickness of the line shows the strength of the association. Here I would like to highlight two key associations. First is the positive correlation between ATP and NAD plus total NAD content. Second is the NAD redox ratio association with the two energy ATP production metabolic rates. And these two associations provide for the first time the direct evidence that NAD indeed play an important role to regulate brain energy metabolism in the brain. In addition, we observe also a trend of a reduction in NAD plus with age. This is actually consistent with results observed also from a study in Minneapolis. So we did not actually observe a linear correlation between the rate constant of ATP production with the age, because the activity of ATP synthase enzyme modulate across the lifespan with a kind of increase from young to mid-age, then a decrease over 65 years old. So we would actually need more subjects in old age beyond 65 to validate this switching point for significant deficit in energy production. So to conclude, our results support the hypothesis that an optimal brain NAD redox ratio and the NAD level sustain ATP production and its tissue levels in the brain. ATP metabolic rate is also a relevant physiological parameter that may help to characterize brain aging. And taking this together, we will say maintaining ATP level and flux via optimization of energy substrates and NAD levels may be a promising therapeutic strategy aiming at preserving brain health and preventing cognitive decline with age, which is the main risk factor for neurodegenerative diseases. So these results also pave the way that we can use phospho-MRS as a powerful tool to support the development of new therapeutic intervention targeting the NAD-associated phosphometabolic pathway in brain aging and neurological diseases. So this is the second application that C13 MRIs will play a leading role to investigate abnormal metabolic processes in a schizophrenia animal model. And so schizophrenia is a complex neurodevelopmental disorder involving gene and environmental interactions. So the proposed hypothesis suggests that the redox dysregulation, neuroinflammation, and abnormal glutamatergic system due to genetic and early life environmental risk factors could contribute to the impairment of parabimine interneurons and white matter in schizophrenia. Ultimately, that will affect the cognitive functions, social functioning, and affective behavior. So this project is aiming to investigate abnormal glutamatergic system using an animal model for schizophrenia. So large amount of proton MRI studies in schizophrenia has pointed to the reduction of frontal glutamate and increase in frontal glutamine. But this result actually provides the average of glutamine levels over different cellular compartments, which is lack of specificity. And it could not provide specific dynamic information such as the glutamate-glutamine cycle and the TCA cycle activity involved in different compartments. So to further investigate whether metabolic activity is altered or which metabolic process involved in the pathophysiology of schizophrenia, so C13 MRIs could be the method of choice in this case. As using C13 MRIs with infusion of C13 labeled substrate, we could be able to assess the metabolic activity related to glutamate and glutamine. In this case, we choose the C2 labeled acetate as it is mainly uptaken by the glial cells and could provide actually more precise measurement of glutamate-glutamine cycle relative to the use of glucose, which is a lack of specificity for the compartment. We used GCM knockout mice and early postnatal oxidative challenge by administration of a dopamine uptake inhibitor to mimic the combined genetic and environmental risk factors. And then we performed the C13 MRS measurement at the postnatal day 30. So before talking about the experimental scheme used in this study, I would like to introduce two main C13 acquisition strategies. We can observe C13 spectra directly from detection in C13 channel. And that offers actually a large chemical shift dispersion and allows the measurement of more C13 labeling information such as level signal in glutamine, also the C4. But this method is suffered from low sensitivity due to the low geomagnetic ratio. Usually, a large voxel will be used. As an alternative, indirect detections through the J-coupled proton nuclear could provide high sensitivity as intrinsically high geomagnetic ratio of the proton. So a small voxel can be used in this case to detect the C13 signals. It also has another advantage. It can provide the direct isotopic enrichment measurement, as the difference and added off spectra can provide total concentration of metabolites and also only the C13 concentration. That allows us to address the isotopic enrichment of different isotope in metabolites. So the disadvantage of this method is the small chemical shift range in proton that limits the number of C13 labeling information we can extract from the spectra. For example, we cannot separate the glutamate-glutamine C2 position even at 14T, but we can still address the C4 position and the C3 position separately. So taking into account the characteristic of the two C13 acquisition method, we could actually, for the first time, using kind of interleave scheme that combine both indirect and the direct C13 detection in this kind of order. And in this case, we can use indirect C13 acquisition, which at the beginning, also across the study, we can acquire the input function of the acetate and also the total metabolite concentration for calculating the fraction enrichment for the direct C13 measurement. And the direct C13 measurement could bring its merit to obtain more C13 labeling time course. And as shown here, we can measure 4 time course lines from both C3 and C4 of glutamate. So input function is the time course actually of substrate isotope enrichment. In this case, is the acetate, and this tells us how fast the substrate actually metabolize in the system, and has to be incorporated in the metabolic modeling to derive the metabolic rates. Usually, it is obtained from measurement using frequent blood sampling. But remember, in this case, we have a small size of these mouse pups, and the blood sampling in these small animals is not feasible. Therefore, using this kind of indirect detection will actually measure real-time input function, solving the problem instead of using blood sampling. So then, using mathematical modeling to fit the obtained C13 labeling time course, we can extract the metabolic flux in the pathway. In this preliminary result, we obtain a reduction trend towards the decrease of glutamate-glutamine cycling and oxidative metabolism. So to conclude, despite the represented by the low isotope enrichment resulting from acetate metabolism and the small brain size in mouse pups, we still successfully demonstrate that using an interleaved approach, the measurement of acetate input function and dynamics glutamate-glutamine labeling and fraction enrichment can be derived without additional blood sampling and in vitro brain extract measurement. And this provides actually the perspectives for studying cerebral energy metabolism during brain development, and also simplified procedure in future human applications. Furthermore, the GCM knockout mice present a trend towards a slower oxidative metabolism and a glutamate-glutamine exchange. But however, with this current number of animals, we could not reach statistical significances, which motivates further investigation in a larger group, larger sample size. So I would like this point to summarize. Overall, multinuclear MRIs is an invaluable tool to investigate complex metabolic processes at a molecular level beyond the scope of MR imaging. Using different nucleus, we can probe specific metabolic pathway and extract the dynamic and static information, including metabolite levels, chemical reaction rates, and the kinetics in a specific pathway. This in turn provides critical mechanistic information that bring in unique insights for studies in brain function and pathology, as shown in the applications mentioned today. Although the field is advancing, but it is still quite underdeveloped compared to MR imaging techniques, which gets a lot of force from vendors. So the main challenge is the low sensitivity of X-nuclear. Some sensitivity enhancement techniques used in high resolution NMR or preclinical MR scanners could not be applicable at a clinical setting due to F deposition regulation and also the long acquisition time. So therefore, we are putting efforts on developing new coils, acquisition, and the quantification strategies aiming at establishing this attractive tool for clinical use. So at the end, I would like to thank a lot of people, our collaborators from Nestle Health Science, especially Bernard Guénon, which is a nice collaborator. We did a lot of scientific discussion and the study plans, and the people from EPFL, Also, our collaborator from SHUV, the Center for Psychiatric Neuroscience, brings a lot of interesting insights from study in schizophrenia. And I also would like to thank the funding support from Nestle, SNF, also Synapse, and the BIG Foundation. And thank you for your attention.
Thank you, Lijing. Thank you for the very interesting talk. At this point, we are open for questions.
Hello, Lijing. François here.
Hello, François.
Thank you for your talk. It was very good. Thank you very much. I just a question concerning your phosphorus aging study. I've seen that the older point had a very large variation from patient or from subject to subject. Did you control on the condition, like if they practiced sport or if they had any diets or things like that?
So actually, we perform this study always at the same time during the day. We are measuring them actually in the fasting states in the morning around 8 to 9 o'clock. So they are arriving more or less with the same condition, fasted states, and without participating in any sports. I would say we didn't really control the daily diet actually, but when they come in the morning, there was no food uptake.
Hello, if I may ask.
Yes, please go ahead.
Yes, so Lijing, very great talk. I come from the School of Life Sciences. You know, I'm doing protein NMR for many people there, and often I see questions that could probably be addressed with things like the kind of things that you presented, but I never know exactly what can be done or not, because I never did imaging or any of the stuff that you do. So on one side, definitely someday we should talk. But now quickly, I wanted to ask you if you can make some parallels and differences between measuring a sample in a small tube and measuring metabolites in your living animal. Because so here, for example, how do you do to select the exact volume where you measure? And then problems like if I prepare a small molecule in solution, it will diffuse very well. So signals will be very sharp. But in your case, the molecules are inside cells, which are very crowded. So probably they will not diffuse well. And that will bring you problems. And then I saw that your points were taken every 20, 30 minutes. Is that because the sensitivity is so low that you have to acquire for a long time? And well, all this kind of, if you can comment briefly, then someday we talk.
Yes. So how we achieved kind of a specific location in the brain, let's say. So this is this what we did with like imaging technique. We do also localization, combination with EPFL and the gradient to select a specific location for the study. And for I think for high resolution, you have the tubes and you don't really specific the location.
You measure everything.
Exactly. So here we do a location. Either you can do with the surface core, like we did for the phosphor. You do the surface core and you dedicate in the sensitive region of the surface core. But you can also combine with localization techniques, like as I just said, using gradient and EPFL, you will be able to select a specific location or a volume you want. And then talking about you were mentioning about the time points, right? The time resolution, and we so here because as I said, the direct C13 measurement has rather low sensitivity, so we were actually able. We have to average for quite a long time, but for the indirect detection, we can actually do more frequent sampling. The actual time resolution is something about 1 minute, okay. And but when we do modeling, we would like to average more time to reach a higher SNR, so we can actually improve the quantification of each time point. But you can also reduce the time resolution, but the reliability of the isotopic instrument quantified from each point will be less reliable compared to more time averaged data. So it's kind of a trade-off you have to balance.
And then a related question about the indirect detection. Can you do something like an HSQC where you detect proton, but you also resolve on carbon?
I think you can. I mean, all these kind of methods you have in the high resolution, we can transfer this to the clinical scale. Preclinical scale. But as I just said, if you want to, the aim is actually to transfer this method to clinical settings, which this kind of method may not be that straightforward because you know this enhancement of signal polarization transfer. You have to incorporate a lot of IF pulses. You know, this brings a lot of star deposition, and it's challenging for clinical settings. So we need.
Okay, thank you.
Yeah.
I will write to you anyway.
Okay.
So
Okay.
you mentioned you're
But
from.
I see the point. Yes. Thank you.
No problem.
Okay. Any other questions? No. Okay. If there are no other questions, thank you again, Lijing.